Okay, now we are going to go on to chapter 6. I'm hoping that the last chapter made a little bit more sense. As you guys can tell from the last two chapters, we are still on the topic of DNA. And just to recap a little bit about what we talked about in chapter 4 to chapter 5, and now into chapter 6. Chapter 4, we were talking about the structure of your chromosome. How is it made? Chromatins, histones, what's involved in creation of the histones? You know, are they an octamer, dimer, trimer? As well as we're kind of getting involved with the idea that thymine and adenine play a very important role in terms of binding and unbinding the DNA from everything else. So whether it's a histone or a chaperone protein or another protein, you're going to look for a lot of TAs, TA base pairing on the nucleotide. Now, from there, from chapter 4, we went into chapter 5, which was all about DNA replication and the idea of mutations and how mutations affect the DNA line versus the RNA line versus the protein line. And this all relates back to the overall central dogma of the cell. So now we're in chapter 6, and we're going to follow the path of the central dogma. We went from DNA to DNA last chapter. Now we're going to go from DNA to RNA to protein. And most of you guys should already have a base understanding about that. If we were to recap the whole entire process, DNA is in the nucleus, RNA needs to be made, and the location that's going to be made is at the nucleus, because that's where the DNA is located. Now, once the RNA is made, as you guys can recall from, your Gen Bio class, either it's Campbell or another textbook, you're going to find that RNA needs to be modified in a eukaryotic organism. We got to, remodify the RNA so it becomes a messenger RNA. But in order to do that, we got to get rid of stuff, add a 5' cap, add a poly A tail, and then fuse them together. Now, once you have the messenger RNA, it's still located in the nucleus, don't forget. Then it gets transported out of the nucleus into the endoplasmic reticulum, or where the endoplasmic reticulum is found. And we then are able to translate that mRNA into a protein. So it's a very logical step, is what I'm trying to say. Now that you have a base path, this textbook is going to go more detailed on how a 5' cap is made. Where does the poly A tail get created? How do we get rid of the stuff that we don't want on the RNA? And then how do we translate once we have the messenger RNA? What proteins are still attached to the RNA? And so let's go through it. So again, we just talked about this. Now remember, this right here, this is all occurring within the nucleus. Okay? It's only when it's ready for translation do we move from the nucleus out into the cytoplasm, where the endoplasmic reticulum is found. Okay? Let me see here. Why isn't my screen working? There we go. And so on your DNA, which is a double-stranded DNA, we have multiple genes. You guys know that, right? Because we just talked about in chapter 5. There's a long strand of genes found on a chromosome. And you could have 10, 100, 1,000 genes, but the rest of the DNA is non-coding. But just know, depending on what the gene is, either we want to create a lot of that gene, we really want to express that gene, which is this path A right here, where we're going to create a whole ton of RNAs, and then all those RNAs are also going to aid in the creation of that specific protein. Or we kind of just need it for one or two things, but we don't really need it fully. Okay? And If that's the case, then that's the path of gene B. So we can say that gene A is a lot more efficient because we're able to create more of gene A protein in the same time frame that gene B is made. So anyways, let's keep going. I don't know why my PowerPoint here is not responding to my... There we go. Okay, whatever. I'm going to use the arrow keys. So again, please make sure that you are reading up on the bullet points here. I feel like since you guys already have the PowerPoints, I'm not going to read through all of them, but they do contain very important like outlines for you to understand this section. Okay, so if you need to have a study guide, this is probably the best way to create your own study guide here, is to copy down the bullet points and see what you know. Now, because we are now in the RNA language, we no longer need the thymine, right? Because we want to be able to have a different set of languages. We don't want to get confused with the DNA. So thymine is no longer going to be a letter that we use. We're going to use the letter uracil, or U. And so know that RNA is ribose nucleic acid, which is a full ribose, which has the alcohol group back on that second carbon. And with that, you have your uracil. Okay? Now remember, you have a phosphodiester linkage when we are creating a RNA molecule. Now diester, again, because there's two ester groups right here on either side, you're going to have two ester groups. So diester with a phosphate in the middle. Now to break these, recall back from chapter 5, to break a phosphodiester bond, we need an enzyme called phosphodiesterase. That's the sole purpose of breaking these phosphate bonds and separating the sugar base from each other. Okay? So again, this is just the difference between the two. Now uracil, again, we're kind of introducing uracil quite often. Last chapter we did cytosine, and when cytosine got deaminated, right, you got rid of the amine group and replaced it with oxygen, it became uracil. Now thymine looks nearly identical to uracil, except it's been demethylated. Now there isn't a process of demethylation that we want you guys to know, but just know the difference between uracil and thymine is the methyl group. Okay, so you can see right here. Now uracil, just like thymine, creates two hydrogen bonds, which is important. Okay? Excuse me. So, now we're getting into RNA synthesis. And in order to make RNA synthesis, we kind of have to take a step back. Now, this slide shows a lot. So let's take a step back. Now, we know from last chapter, DNA synthesis requires an enzyme called DNA polymerase. And RNA, to synthesize RNA, we also need an RNA polymerase. Now, the question that your textbook goes over is, are these going to be the same protein? Will they have the same behavior, the same shape, the same configuration? And researchers have pondered this for a long, long time. And really, it all comes down to, do we assume that they're the same or not? And with studies that's been shown in the past decade or so, several decades actually, we've discovered, or scientists have discovered, that RNA polymerase is not the same as DNA polymerase in terms of what's holding it together, the shape of it, the functionality of it. It's similar to DNA polymerase, but it's not identical. We cannot overlay the two. You know, some enzymes, they're very similar. We can overlay it, and you'll see that the folding of the protein and the active site of that protein pretty much is identical. But for RNA polymerase and DNA polymerase, that is not the case. And so what we have right here is an RNA polymerase, okay? And we can see that the RNA is trying to transcribe the DNA. Now, one thing that I do want you guys to take note, right here, we see that the DNA, the double-stranded DNA in yellow, is bent. And the reason why it's bent is to try to get one of that little strand open. A good way to think about it is like when you're going to the grocery store and you have plastic bags and you tie them in a knot to make sure everything doesn't spill out. And now, because you've tied it as a double knot, how do I untie it? Well, you got to take one of the strand out and kind of loosen it from the other, you know, and then pull it apart. And the RNA polymerase, very similar to the DNA polymerase, needs to be able to do that. So it's going to have helper proteins bend this DNA, maybe trying to wedge itself in the helix to kind of untie it and then grab a loose piece of the DNA and pull it out. That's number one. Number two, notice we are now having a very specific cation involved. This is a magnesium ion. Now, we haven't talked about nutrition because this is not a class of nutrition. But you guys know that when you take your daily vitamins, if you read the nutrition label, there are some minerals involved. And these are your microminerals. These are the micronutrients that you need to be able to efficiently work at optimum levels. Imagine if this magnesium ion isn't here. Well, what's the purpose of the magnesium ion? Remember, it's a charged atom. It's charged. What's inside of active site? A lot of R groups on the amino acid. Now, some of these R groups are going to be polar. Some of them will have an electrostatic interaction. Some of them might have sulfur bonds involved. And in this active site with the catalytic reactions, you know, this magnesium is going to play a role. One of its role might be to hold everything together, right? All the electrostatic interaction is going to permanently bind to this magnesium, holding the shape of this active site so that it doesn't fall apart. It could also be used, you know, maybe hydrogen bonds need to find a way to kind of hold itself together, and this is indirectly holding it together. Who knows? Okay. But the point is, you have all these micronutrients, copper, magnesium, iron, that you need to consume. And the reason why we need to consume it is because sometimes those ions are very specific in holding your proteins together that will give the overall enzyme a overarching functional action, function, whatever you want to call it. Okay, I'm at a loss for words right now. So this is essentially what I want you guys to take from this image. Now, there is a description. Please read it. But know that we are still synthesizing, just like DNA polymerase, we're still synthesizing from 5' to 3'. We're reading the DNA, if you guys can take a look, from 3 to 5. Okay. Now, this image is an amazing image because we can see the DNA right dab in the middle. The DNA is completely highlighted in the center right here, and we can see all these branches veering from it. Now, this is an electron micrograph or electron microscope image, and what we can see is that on these specific areas of the DNA, aka the genes, so you can see this might be gene A right here, and this is gene B, we can see that the protruding bits are all RNAs. We're creating RNAs. But look at it. We're not creating one RNA at a time. We want to be efficient. We want to make 1,000 RNAs in a time frame of an hour. We can't do that if we go one by one by one by one. What's going to happen is that one's going to start, it's going to go down a couple nucleotides, and then another one's going to start, and then another one, and then another one. Now, here is a nice question. Which direction is the DNA? if I was to ask you guys to label 5' to 3' or 3' to 5' or 3' this direction 5', yeah, which way is the DNA and how do we tell? So take a moment, really take a look at this image and just see which way is the DNA facing, 5 to 3 or 3 to 5. Assuming that this is a single-stranded DNA that RNA is being transcribed on. Okay, which direction are we going? Now, you guys might know this already, but if you don't, take a look. Where is the RNA starting? Well, how do I tell? Well, when an RNA polymerase just begins to transcribe, it doesn't have an RNA attached to it. So, look at the site where it's not attached. Well, it's right here at the tip of the Christmas tree. That looks like a pine tree, right? This is the tip right here. This is the starting point. As it's transcribing the DNA, the RNA should get longer. So as we go down this chain, look at the RNA. The RNA is extending. What direction does RNA polymerase synthesize in? 5 to 3. That means which direction is the DNA? Well, if I'm synthesizing from a 5' to 3' direction, I need to read the DNA. Remember, I need to read the DNA in 3 to 5. So my 3' is right here on the DNA, and my 5' is right here on the DNA. So this is a 5 to 3. 5' starting on the left, 3' starting on the right. And how I could tell is by looking at where the RNA is starting. This is a great test question because it tests you guys on directionality. Where's the DNA starting? Where's the RNA starting? What happens when we start transcribing an RNA? This is great. Okay, let's move. All right, so now for our textbook, we go over both prokaryotic and eukaryotic transcription. Now, for prokaryotic transcription, it's very simple. We know that the DNA is circular, right? Because in the bacteria, it's very small. And so we need to find a way to start transcribing. Now, for both cases, you need a start region, right? To start any process, you got to have a region where you're going to begin. And this is what we call a promoter region. So somehow on the DNA of the bacterial cell, there should be a promoter region, a very specific set of sequences that when an enzyme or a factor, transcription factor, floats around, it's going to be able to identify that extremely specific sequence and latch onto it. And once that sequence latches on, or once that enzyme / helper protein latches on, then the whole process can start. But you need that promoter region. Now, for eukaryotic cells, we have a promoter region where there is a start, but we also have additional region like the TATA box, the INF box, the other regions where other promoters or other transcription factors will attach to start the whole process. And we'll get to that in a minute. But let's keep things simple here. This is a bacterial cell. So the transcription factor that a bacterial cell uses is very simple. There's only one, and it's called a sigma factor. So for prokaryotic cell, the sigma factor is the transcription factor for the RNA transcription process. And what's going to happen is that the sigma factor needs to interact with the RNA polymerase. Now, once you create this complex right here, this is what we call a holoenzyme. Okay? And it's going to bind, this whole complex is going to bind onto the double-stranded DNA. And you can see right here, it's latching on to a good sizable amount of nucleotides right here. But in step number 1, what happens is that one sigma factor, one end of the sigma factor is on one end of the DNA, the other one's on the other end, and then somehow it's going to untwist, bend, and expose one of that DNA strand. Now, once this is exposed, then the polymerase can come in and start yanking the yellow strand out, right? It can yank the yellow strand. The red strand stays because that's just a complementary strand. The template strand, which is the yellow, is going to get yanked by the polymerase into its active site, and it's going to start the warming up phase. Now, I call this a warming up phase. This is called the initiation step. Either one. Initiation step is the textbook way, so I recommend doing that. But for concept-wise, let's just think about it as warming up. And what's going to happen is that the open complex is going to transcribe. Now, it's going to transcribe the first set of nucleotides right here once, and then eject it. And then it's going to rewind the DNA back into itself and transcribe it one more time. Eject that, pull it back, transcribe it one more time, eject it, and then pull it back and transcribe it one more time. So you can think of this like starting a gas lawnmower when you are cranking or yanking the cord. You yank it a couple of times, it doesn't start, and then finally at the last yank, then the machine starts. That's essentially what's happening here. It's going to yank a couple times, concept-wise, right? It's going to yank it a couple times. It's not going to like it. And then finally at the final yank, then the engine runs and then the polymerase can do its thing. Okay. Now we want to be able to create these for reasons. Number one, we want to make sure we have the nucleotides around. Okay. All the triphosphates around to be able to work with it. And, you know, as you're warming up, you're going to make some errors. So these might have errors on it. So we're going to do it a couple times just to make sure it's kind of working. The polymerase is working correctly. Fine. And then let it go once we find it's working. Once the RNA starts building, okay, once it starts building, this sigma factor gets dislodged because now the polymerase is working. The sigma factor no longer needs to be attached to the DNA anymore. Again, the role of the sigma factor is to bend and open the DNA so that the polymerase can work. But once the polymerase is working and the strand is already unwound, sigma factor doesn't need to be there. It's going to dissociate separately and then bind to a brand new polymerase enzyme and then run the process again. Now, with the original polymerase right here, so once you clear the promoter region, so this is when the sigma factor gets released, is after we transcribe the full promoter region, the sigma factor gets released. The RNA then gets elongated. And remember, this is a single-stranded RNA. Single-stranded RNA has exposed nucleotides where it can form a hydrogen bond. What's going to happen is that as it's being created and as the strand gets longer, you can see it's going to twist up on itself, fold in on itself like a protein would, and create a hairpin shape. Now, once you have this hairpin formation and it gets a little too strenuous, what's going to happen? It's going to get yanked out of the polymerase. And then once that's happened, termination ends, right? The termination process has finished. So now you have a fully folded RNA molecule, not mRNA, RNA. You have your double-stranded DNA, and then your polymerase. That's now dissociated. And now this polymerase can bind with another sigma factor from another complex that just got released, and then start the whole process all over again. So it's a very simple 6 steps, 6 to 8 steps, okay? But if you think about it just in a very simplistic sense, you know, I'm hoping that it's kind of very reasonable on the actions that these enzymes will take. Okay. Now, with all the bacteria and all the studies that we've done, we've created a chart on all the promoter regions, at least 300 promoter regions, and compare the nucleotides found at that promoter region. And what we found is a consensus sequence. Now, if we take a look at this chart right here, we can see that this is 35 nucleotides long, 10 nucleotide sequence, 35 nucleotide sequence. And we can see which letters, which nucleotide is most prevalent at that specific sequence out of the 300 that we've studied. And we can see for the majority of the time, we're going to have thymine on the DNA with another thymine, with another guanine, with another adenine, with the cytosine and adenine. So this is a very conserved sequence in a lot of the promoter regions for E. coli. So it would make sense for a transcription factor / polymerase to have a recognition site where it can recognize TTGACA for most of its transcription. Right? And same thing with TATATTA, Tatabox, 10 nucleotides long. Okay? So, let's keep going. Now, we know that DNA carries a lot of nucleotides. We know DNA with those many pairs of nucleotides creates several genes. However, we don't want all the genes to be only found on the template strand because either strand can be a template strand. Okay? A lot of students will think that when we draw a DNA strand, one has to be the template, one has to be complement. That is true when we are doing DNA replication. But remember, these are identical strands from each other. And sometimes one gene is found on one strand and the other gene is found in the opposing direction of the other strand. This makes for efficiency. We can pack more information in the DNA if we are able to kind of utilize both and understand the general direction of the DNA. So we can see right here that these green triangular bits, these are your promoter regions. Now, depending on which direction the DNA is going, right? So this is the promoter region. The DNA is going from 3 to 5, and we're synthesizing from 5 to 3. That means from this point right here to this point right here on gene E, this is one gene. Your gene F, is on this promoter region to here. Notice they're very close to each other, but we don't want them all to go in one direction because, again, if they're all going in one direction, all the RNA is going to cram up on one side. RNA polymerase is going to cram up on one side, and it's going to be very inefficient. So we want to have transcription be at both directions for efficiency's sake. Okay? Now, that is prokaryotic transcription. Now we're going to talk about eukaryotic transcription. Now, for eukaryotic transcription, we have 3 RNA polymerase. Now, remember, eukaryotic cells are more complex. They are magnified 3x, 5x, 100x times that of a prokaryotic cell. So with more complexity, you need more tools. So RNA polymerase 1, 2, and 3 are those tools. However, for our purpose, for our class, we are going to be mainly focusing on polymerase 2 because only polymerase 2, RNA polymerase 2, codes for protein. Polymerase 1 and 3 codes for small RNAs or ribosomal RNAs or tRNAs. But it's only polymerase 2 that's involved in the creation of protein. So, how does it start? Well, just like eukaryotic cells, you need to have transcription factors. You need to have helpers to open up that DNA or to show the polymerase this is where you need to start. Now, notice we have a Tata box. Again, a region where it has a lot of thymine and adenine. And usually, this Tata box will be located around 30-ish nucleotides above where you want to start transcribing. Now, it's really specific on the spacing. Why is that? Because your transcription factors are going to bind to this spot first. And then your polymerase is going to interact with it and might be shifted downwards where this is the true starting point. You got to give it some room so that when it binds to transcription factors, there is a buffer region so that nothing is left out, in essence. Okay? So, what recognizes this Tata box? Well, it's your first transcription factor, called the Tata binding protein, the TBD. Now, your TATA binding protein forms a complex with your TF2D, aka your transcription factor 2D. Okay? So once you have this complex here, this transcription or this TATA binding protein is going to latch onto the TATA box, opening that DNA. The transcription factor 2D forms that complex, wraps itself around the DNA, and then transcription factor 2B and A comes in to kind of fully synthesize that complex, where it holds one tail of the TF2D on one end, and then you can see the TF2B holds the other end right here. Now that we have the initiating complex, notice what happened to the DNA. Your TBD latches on, pinches your TATA box. All the other transcription 2 factors are bending the DNA, very similar to the sigma factor. We bend the DNA to expose a single strand of DNA. Now, once that happens, then your polymerase, remember, we're transcribing protein here. We're going to focus on RNA polymerase 2. Your RNA polymerase 2 comes in and inserts itself into your transcription factor 2 complex here. Okay? And once your polymerase comes in, then transcription factor 2 H, E, and F integrates with the polymerase to form the overall complex. Now, one thing to note, CTD, remember, this is a polymerase. NCC, NCC, NCC for proteins, remember that, right? This is all the way back in chapter 3, NCC, NCC, NCC. On a protein, you have an N-terminal N, and let me get my pencil out. You have an N-terminal, right, that starts all amino acid sequence, and you have a C-terminal, okay? Now, if you guys recall, when we were making histones and all of that, all the N-terminals, where were they? Were they inside or outside? Right? They were outside because they were able to get methylated. They were able to get altered on the N-terminal end. They were sticking out. Do not get that confused with polymerases. Histones are slightly different. Polymerases, they have a C-terminal domain. Now, this domain is very important. This is going to be where other enzymes or proteins are going to latch onto. We're going to phosphorylate this N. We're going to, modify this C-terminal domain N to activate the polymerase. It's like putting a key into an ignition hole, right? Into a keyhole. You're going to turn the key in the keyhole and then start the engine. So same idea here. The C-terminal N is going to stick out, waiting for other enzymes to latch on. And later on, as we transcribe the RNA, what's going to happen is that as the RNA comes out, these proteins will have a stronger affinity for the RNA, latch onto it, and begin modifying the RNA so it turns into an mRNA. But in this case, for the C-terminal domain right here, okay, just know that, let me see if I can word this in a simple way. Give me a moment. Actually, the C-terminal domain also acts like not only an ignition, but it will act like a, what do you call it? A switch. No, switch. Yeah, acts like a switch. Because once everything begins to transcribe, we can also modify this C-terminal domain where it turns itself on, the polymerase runs, and remember, like sigma factors, we don't want all these transcription factors to stay on the polymerase. Because once a polymerase is going, we can use the transcription factor for another polymerase that needs to get started. Does that make sense? I hope it does. So, because of that, all these transcription factors, once the polymerase gets started, will pop off. Okay. So, if we take a look right here, You have all the transcription factors attached, okay? UTP, ATP, CTP, GTP. Remember, these are your nucleotide triphosphates. This is the free form of nucleotides that is floating around in your nucleus. You also have TTP for DNA, okay? Polymerase takes the triphosphates, gets rid of a pyrophosphate, and there is a nucleotide monophosphate attached on the nucleotide chain. Okay, so don't forget that we still have, AMPs, CMPs, UMPs, GMPs while the RNA is being created. Now, once all of this gets initiated, then elongation transcription factors, which is going to attach itself to the polymerase, modifying its shape slightly so that it can ride along the DNA and begin transcribing. Notice the polymerase is now dislodging itself from transcription factor 2. But transcription factor 2 isn't going to lop off, right? Remember, in step 1, it took a while for transcription factor to prep this. And once this unit has been made, then a polymerase comes in. So once a polymerase leaves, another polymerase is going to come in and lock itself into here to start again. And if you recall from our image prior, where is it? Let me show you. We have multiple polymerases happening at once because the transcription factor here is still locked into the DNA. So all we really need to do is lock that structure there. Polymerase comes in, starts transcribing. All right, another polymerase comes in, starts transcribing. Another polymerase comes in, starts transcribing. Okay? Now, here is a chart that tells you exactly what everything is doing. TF2D has 12 subunits. It recognizes TATA box. TF2B recognizes the BRE element. TF2A it's not required in all promoters, but it helps stabilizes the shape. So these are things that, again, holds the structures together. Now, they're introducing BREs, C-terminal domains. They're introducing a lot of new extra words. But just know that your TATA box, your INRs, your DPEs, your BREs, these are all here for your transcription factors to bind to. Remember, we got to keep the structure stable. So you're going to have a transcription factor. Let me pull my pen out. Bind to the BRE. You're going to have transcription factor 2 bind to the TATA. You're going to have transcription factor the 2D also latch onto the TATA because it's a huge subunit, right? So these are the two regions it's going to anchor itself into, but it can't anchor itself into this region unless it's able to read the INR sequence, which is usually these nucleotides here. Okay. Now, if you see the letter N, that's for any nucleotide. It could be A, T, C, or G. But it will generally have this region, and TF2D can recognize this specific region to latch onto, as well as DPE. Okay? Remember, transcription factor 2 is huge. Look at how big, right? It's covering all of this space right here. So you have the BRE site right here. You have your TATA site right here. You might have your INRE right here, and then one more right here down the road. But all these 4 regions help the TF2 wrap itself around the single-stranded DNA, or the DNA currently, and anchor itself. Okay? And then right here is just an example of what happens when we bend the DNA. You can see the kink forming, and you can see the exposed red or yellow single strand. Okay. Now, with this, and say that we want to really ensure that the whole complex doesn't move anywhere. Okay. We need an initiator, which is your promoter sequence, but we also want to keep things there if we want to really transcribe a lot of RNA. And so if we want to really transcribe a lot of RNA, not just one, but we want to transcribe 1,000, we need another piece to hold everything together. Now, this is what we call an activator protein. We activate the whole complex so that it stays on. So an activator protein will bind to an enhancer region, which is well in front of the TATA box, okay? Well in front of, just say the TATA box for now. And it's going to bind here. Now, this activator protein will have a very specific conformation. Reason being is because we're going to have a new enzyme come in called a mediator enzyme, okay? And it's going to bind itself to the overall complex of your transcription factor. Now, to keep the mediator on here, this enhancer protein is going to bend the DNA, wrap itself around, and lodge itself into the mediator protein. So we can see that right here. We can see the DNA bend, and the activator protein is now solidifying its shape right here so that the mediator protein doesn't move left or right. It stays put. And then once this happens, now transcription can just be on autopilot, right? Because nothing's getting shifted around. The main polymerase 2 and transcription factor 2 aren't going to be shifted around too much. We can run the activity. Now, just know that this picture only shows one activator protein. But again, in a real world situation, we want to be as efficient as possible. There's going to be multiple activator proteins holding this thing together. Okay? Could be at different regions of the DNA. It might hold at different sites of the mediator protein, but it's going to be multiple. And with that, testing. Hello. Okay. Sorry, guys. My microphone kind of cut out really quickly. And with that, that is the start of eukaryotic. What is it? Transcription. Okay. Again, the whole point of the mediator is to hold everything together. Once it's together at the promoter region, then transcription can continuously go. Okay. So, going back to what we did at the very beginning of the slide, we have, let me see here, we have the difference between a bacterial cell and a eukaryotic cell. We know that bacterial cell, once it's transcribed its DNA into RNA, it is immediately an mRNA. No modification required. Why? It's simple. It's a simple organism. It doesn't need to do any modifications. However, for our purpose, for our eukaryotic cells, we need modifications. We have introns and exons. We got to get rid of the introns and splice the exons together. We got to add an mRNA 5' cap, and we got to add a poly A tail, which we call polyadenylation. And once all of that is together, then we got to transport this whole molecule out through a nuclear pore into the cytoplasm, and then we can start translation. Okay? Now, I am going fairly slow because this is a lot. If you guys want, speed up the video 2x so that you guys can get this at double the speed. But there's a lot of things to break down here. And I wish chapter 6 was shorter, but it's not. Okay? Now, this is what bacteria undergo and what a not poly A tail, what a 5' cap looks like. So in figure B right here, your 5' cap, you can see there's several nucleotides, right? And we can also see a methylguanosine right here. Okay, but notice that it is a 5' to 5' phosphate connection, triphosphate connection. So your methylguanosine, aka MGTP, right, is latched onto the 5' end of your RNA in the 5' direction. So it's a 5 to 5. So this prevents any other polymerase coming in to read the beginning of your RNA and cause a malfunction, right? So this is a cap to stop things from interacting with the 5' end. So you can see the guanine with the methyl group attached and a triphosphate. That is your 5' cap. Okay, your poly A tails is just 150 to 250 nucleotides long of adenosine. Okay, now you're going to have coding sequence and non-coding sequence. Obviously, your coding sequence is going to be the middle piece, and your non-coding sequence, because we don't want to damage the genes, okay, is going to be at the end, and then hopefully we can make protein out of it. So, what happens here? Well, in this process right here, we know that this is a polymerase. You have a 5' end of your mRNA starting to transcribe right now. It's beginning. Now, once it gets long enough, okay, you're going to have some splicing protein right here, okay, come on in and latch itself on to the RNA. Okay. Now, this splicing protein is going to be very important because this is what aids in the splicing of exons together. But notice, what I want you guys to notice is notice where they are found. They're found on the C terminal domain. Now, there's also a capping protein that is first there. So once the 5' end gets long enough, this capping protein is going to encapsulate the RNA to protect the tip, and then as it gets longer, more splicing protein is going to be latched onto it, because again, it's going to aid in the excision of the introns. So how does a 5' cap work? Well, right here we have the 5' end, we have triphosphate with some sort of nucleotide with another phosphate nucleotide. So at the beginning of your 5' end, you're going to have 3 phosphates. Great. Well, what's going to happen is that as the RNA comes out, one phosphate group is going to leave. It's going to get dephosphorylated. Okay. And then you're going to be left with a diphosphate end. Now, we don't want diphosphate. We want triphosphate because that's the 5' cap. But we also want that triphosphate to be a guanine. Because remember, it's a methylguanosine 5' triphosphate. So what happens? Well, we have one phosphate group right here, and we can attach GTP, or guanine triphosphate, onto this 5-terminal end. Now, we don't want to add all 3 phosphates onto it. So what's going to happen is that we're going to have GTP. Okay, so here's G, here's P with 3 phosphates, right? We're going to splice out the pyrophosphate, or cut out, slice out the pyrophosphate, have the GP attached onto here, and the pyrophosphate leaves. Now, once that happens, now you have your guanine phosphate attached with the other 2 phosphate that was originally on the 5' end. And now we got to methylate it. So a methylation process occurs somehow. Some enzyme will come in to methylate it. And we're going to methylate, adding a methyl group to the guanine, giving it a positive cation charge. And then once that happens, we want to stabilize the whole cap. We're going to methylate the first nucleotide that's in contact with the guanine. And then once both are there, now you have a stabilized 5' cap. Okay. So 2 methyl groups are required to stabilize this 5' cap. One phosphate nucleotide, a GMP, is needed to begin the capping or begin the structure of the capping. Phosphate obviously has to leave first. Okay, so now here is an interesting concept: the splicing of the introns and exons. Okay, this is going to be a two-part video, unfortunately. I'm going to stop once we get to translation, but this is about to hit translation. In order to really cut out an intron, couple things that we need. Notice we need a U1, U2 complex. Now, this complex is fully RNA molecules. We call these snRNAs, small nuclear RNAs. Okay? Now, these are responsible for maintaining the stability of your RNA, but it's also responsible for latching on to the intron. And then when we splice it, we're going to create a brand new structure while having the exons fuse together, and then the introns fly away with all of our spliceosomes or splicing factors. Okay, so right here we have this highlighted A. Now, this highlighted A is not a adenine. It's actually the branching point. If you guys can see, it's the branching point where we can cut off the intron. Okay, so it's going to branch off in this direction. We're going to cut it somewhere right here, cut it right here, and then introns will leave. Okay, so note we have a lot of introns and exons, but more introns than exons. Introns have a bigger, thicker region. And depending on where we're splicing and what introns and exons we keep or remove will determine what type of protein we make. Now, to keep things simple, what is happening right here is that as we want to remove an intron, we can see the branching point right here is going to pretty much fuse back together where the beginning of the intron is, separate itself. Okay, you're going to have a free alcohol group right here, recombining back to the next exon. This is the intron right here. Okay, and it's going to essentially get cleaved off, forming a shape called a lariat. A lariat being a circular loop. And then you have your completed exon sequence here. Now, the structure of the lariat is structure B right here, or column B. So this is what a lariat will look like. You have a 5' end of the intron attached to the 3' end of the intron over here. The new bond that's forming is a phosphate bond. Okay? But that's essentially what the lariat looks like at its branch point. Okay? Now, if you want to look at the splicing of the gel through electrophoresis, remember, depending on how large it gets, it will determine how far down, or how small it gets, it will determine how far down it goes. And we can basically see the different types of splicing fragments throughout the cell. And we can stop it at 1 minute, we can stop it at 2 minutes, 3 minutes, 4 minutes. But what you can see right here is at 1 minute, we have just one massive molecule. We don't know what it is. It hasn't begun splicing yet. At 2 minutes, we've started splicing or started cutting it apart. So right here, at 3 minutes, we have a couple more, but not as big as the original, but it is getting further down. Okay. And then at minute 4, we still have the long strain right here, but nothing down here. So what's happening? Well, remember, at time 0, we have a long mRNA strand. At time 1, we are starting to cut that strand apart and fusing them. Now, some pieces might still be there, some might not. We don't know yet, but we got to wait, right? So that's why we have multiple fragments at time 2. At time 3, now we have a whole bunch of lariats forming, as well as a more synthesized RNA, mRNA, okay, which are these two right here, okay, and at time 4, the RNA is no longer present. Why? Because it left the nucleus. There's nothing there. Okay. So again, these are the labels right here that you guys can see on lane 1, 2, 3, 4. Okay. And your textbook shows you this picture. Now, again, all I really want you guys to take away from this picture is depending on where your protein's getting manufactured will determine what genes are spliced together or what is left in the nucleus. So you can see this is the same gene in 5 different locations. Okay, tropomyosin gene in 5 different locations. You have the striated muscle, the smooth, the fibroblast, another fibroblast, as well as the brain. But if you take a look at where the splicing region is, and then where we're going to cleave, right? These arrows are where we also cleave. You can see that this blue region is this first exon right here. This blue region right here is this exon, if we line it up, versus in the smooth one, this is the weird one, we keep this exon, not this one. And then you go down the line, and depending on what exons you want to keep, will determine what type of tropomyosin you're going to create. Because again, depending on the location of the cell, you want to have the structure most suitable for it. Okay. So, how does splicing work? Well, take a look right here. You have the single or the small nuclear ribosome, okay? But the SNRPs right here, SNRNP, forms a base pair with the 5', so it's a nuclear protein. Okay, it's going to bind itself so that the small RNAs can come in and work its magic. But once you have that first SNRP, U1 SNRP right here, U2 is also going to SNRP. But don't forget that we need the branch binding protein right here. Branch point binding protein. The branch point binding protein is going to bind itself to the adenine and other nucleotides attached to it. And it's going to get stabilized by the U2AF. Now, once you have these two regions here, okay, you have another SNRP protein coming in. You have U2AF is going to leave because now we have a complex latched onto this. But we need energy for this process. So ATP is going to get involved. So you're going to phosphorylate, you're going to bind itself, and then what's going to happen is that now you have 2 enzymes, or 2 complexes here, RNA complexes here, that is going to essentially maneuver itself where it's going to get closer and closer together. Notice that more helper structures are coming in, okay? They're going to bind itself to create a overall complex here, where all of them interact with each other. And then this creates an NTC-NTR protein complex. Now again, this requires a lot of energy. So you're going to have the ATPs involved. Once you create that complex, now the active site is ready to begin its catalytic reaction. It's going to start cutting covalent bonds. It's going to start reattaching or phosphorylating regions, maybe dehydrate, right? It's going to undergo dehydration, okay, reaction. And what's going to happen is that your laureate is going to get formed. We're going to cleave off, forming the alcohol group on the 3' end of the 5-terminal piece, and then fuse it together on the 5' end on the 3-terminal piece. And then once that happens, then you have a spliced RNA. Now, one thing to note is that all these proteins and RNAs that are involved in the splicing is what we call a spliceosome. So just like a nucleosome, where it's histone proteins with the DNA, or replisomes, where is replicative enzymes with the DNA, a spliceosome is splicing enzymes and RNA with the mature RNA, mRNA, or the creation of the mRNA. Okay. So here's just a zoomed in interpretation. And we can see essentially what is happening. And don't forget, you still have magnesium involved. Not only is magnesium involved in the polymerase 2, but it's also involved in the spliceosome activity. Okay? And then here's another micro electron graph. Okay, micrograph. Okay, you can see the spliceosome complex right here with the Lariats forming. So you can see the Lariats here. Yeah. Okay. Now again, which direction are we looking at? This is a great question, right? Where is the 5' direction? Where's the 3' direction of the DNA? Well, we can take a look. Or, yeah, DNA. It should be DNA, right? Again, these long strains are just regular RNA. It's regular RNA. We're still in the nucleus. This is your DNA strand. Okay, this is your DNA strand. You still have RNA being created. But again, we want to be as efficient as possible. So once the RNA is created, what happens? We want to transfer that RNA or recreate that RNA to become a mature RNA. Hence, your Lariats are forming. Remember, we're synthesizing from 5' to 3'. We're reading the DNA from 3 to 5. That means this is your 3' end. This here is your 5' end. Why is that? Well, again, you have enzymes attached to the DNA right here. Nothing's happening. And then all of a sudden, RNA starts forming. And then it's starting to get longer as we go down the chain. So, if it gets long, that means it's transcribing. And if it's transcribing, we're reading from 3 to 5. Last but not least, so we talked about the methylation and the 5' cap. We also have the poly A tail. So again, we cleave off around 30 nucleotides-ish away. We're going to cleave this off where it contains a lot of guanine and uracil. Okay, this will then get degraded in the nucleus. And then a poly A, which is all A's, adenine, will get attached to the very end. Okay, now we use a term called a hexamer. And when we're using the term hexamer, it's not the same as like an octamer or a dimer. Hexamer is actually related to how many nucleotides we are looking at. Remember, mer represents unit. Okay? It's the suffix, or it's the suffix representing unit. So a hexamer is just something that has 6 units on it. Okay? In this case, when we're talking about a hexamer, because we're mainly focusing on nucleotides, we are looking at 6 letter combinations. Okay? So, how is a poly A tail created? Well, again, that C-terminal domain, the CTD right here, is important, like I told you guys. You're going to have enzymes attached to that C-terminal domain, and as your RNA gets matured, what's happening is that cleavage and the poly A signal is encoded in the DNA. So you got to read a signal. Hey, this is when the RNA stops. All right, polymerase is reading that. Great. Now we need some cleavage factors. So these right here are your cleavage proteins. Okay, they're going to bind to that hexamer coding, AAUAAA. Now, once you have this hexamer coding, okay, more factors are going to come in, and it's going to cleave the rest of the RNA out. Now, because we have an open end, a poly A polymerase comes in and latches itself on to the CPSF binding area. And then it's going to start polymerizing. Now remember, these are all adenines. They're all adenines. and they're going to get exposed, and we don't want them to get damaged while we are making them. So we got to have something that protects them. And this is where your poly A binding proteins come in, your PPAPs, okay, or PAPs. And once they're in, they're going to shield the poly A tail so that it can exit out of the nucleus through the nuclear pore. Okay, so here's what a nuclear pore looks like. You have a central channel right here, and it should be just big enough so that things can move in and out, mainly your mRNA. So again, here's your DNA, your chromatin structure. You have an RNA being formed from the RNA polymerase. It gets longer, and then all of a sudden it gets reconfigured. Okay, has some modifications onto it. You have the 5' cap made. A lot of the proteins are still attached to it, and then all of a sudden it gets cleaved off. You have your poly A tail now still wrapped around with more poly A binding proteins. So you have now a cluster of proteins. Now to export it out properly, what's going to happen is that these proteins, depending on what type of proteins they are, either stays attached to the mRNA and then lodge itself into the endoplasmic reticulum, or they get dissociated and stays in the nucleus to start the process all over again. Okay, we don't want to get rid of nuclear proteins that we need to use again. It doesn't make any sense. So what happens? Well, here's your complete mRNA. Okay, so this is your complete mRNA. You notice all the proteins attached to the mRNA right now. We got to protect it, right? It's going to flow out, but these are your nucleus-resistant protein. They need to stay within the nucleus. So what happens is that as it's going out, you have a nuclear export receptor here. It's going to latch itself on to open the gate. Okay, so here's the pore. It's going to open the gate. So this receptor is going to latch itself on right here, opening the gate. And then the tip of the mRNA, the 5' cap, is going to go through first. And as it's going through, again, the pore can only fit so much of the mRNA through that anything that's outside of that range is going to pretty much get dissociated. So all the nucleus-resistant protein stays inside the nucleus. It's going to come out. Now it has exposed ends and other receptors here. Protein synthesis factors now come in and lodges itself onto the mRNA. Okay. Once it lodges itself onto mRNA, the 5' cap, or CBC, now gets dissociated, and now you have translation that's about to occur. This is your fully matured mRNA ready to undergo translation. Okay. So So I would recommend at this point in time, pause the video. I'm actually going to create one solid video, but I recommend you guys pausing this. This is a lot of information. We went through quite a number of detailed steps just to go from protein to DNA to RNA, and then from RNA out into the cytoplasm. I recommend reviewing your steps from here before continuing on, because once we continue on, if you remember the background of your translation, most of this will be a lot easier to grasp. Okay, so just fast forward a couple slides, we're going to talk about your amino acid codon box. Okay, this is your codon box. MET right here is your start codon, right? So do you guys remember this from GenBio, I hope? If I gave you guys a sequence of amino acids or of codons, hopefully you guys will tell me what the amino acid sequence are. Okay. So I recommend stopping here, take a break, and then go on. Okay? So take a break.